Hello everyone, welcome to this PalmSens webinar. I'm Lutz Stratmann. I would like to start maybe by telling you who is going to host this webinar today. Well, I already introduced myself, but I also would like to introduce somebody who you might already know if you have attended multiple of our webinars, which is my colleague Ardi. There we go. He's going to be your voice. He's going to be the one that is translating your questions to us. So please, if you do have questions during the webinar, feel free to write them in the chat. But actually, it would be even more handy if you use the Q&A function of Zoom, where you can write in questions and people can actually upvote them, so we see which questions people are the most interested in. And I already told you guys that there will be a Q&A this way at the end of this webinar. So you will have time to ask questions. Yes, and who will you ask the question to? Well, that is of course our star guest. We have invited somebody who's not from PalmSens. It's actually Marcin Szymon Filipiak, who is assistant professor in at the Institute of Physical Chemistry in Warsaw. Marcin, maybe give the audience a wave so they see who you are.
Hello, everybody.
Very well. So first I will tell you, like, how did we get here? Why is this happening? And then Marcin will take over and tell you a lot about field effect transistors, how to use them in sensors. And oh yes, as a reminder, this is part one. We're going to have a second part next week that will, from the structure, work similar, but the topics will be more advanced, but more about that in the presentations. Most of you most likely know PalmSens as a producer for portable electrochemistry. That means we're making potentiostat, galvanostats, and devices for impedance spectroscopy, and we always try to focus them on small sizes, battery, Bluetooth, so that you can do electrochemistry everywhere, be it in a very crowded lab, be it at your project partner's lab, or out in the field. Right. And also, multiple of our devices are actually bipotentiostats, meaning they have two working electrodes that they share with the same reference encounter electrode. I made the green check marks at the devices here that you see that are already available as bipotentiostats, like the PalmSens 4, the Sensit BT, or the EmStat Pico. And over the years, every now and then, we had the question, well, can you use these for field effect transistors? And we were like, well, we think, wait. So we thought, yes, it must be possible, right? But we've never tested it, and that was not good. But we thought, okay, let's look into literature. And we were a bit surprised how little documentation there is out there, how to use let's say, your everyday potentiostat for field effect transistors, because they are a part of the kind of the electrochemistry community. Well, there's a bit of discourse there as well. Anyway, so we thought, okay, we should really start a project to see how this is working. And we wanted to focus in that. So the project was, do our potentiostats work as characterization tool for field effect transistors? And we did start doing that with the EmStat Pico, right? So this is the small potentiostat module that you see here, which is capable of impedance spectroscopy. It's also the foundation for the Sensit Smart and Sensit BT. Basically, this module that you see in the middle is in the devices that you see on the left and right. And it is a bipotentiostat, and we thought, okay, with that size, and already a bipotentiostat, it would be great if it is suitable for field effect transistors. Plus, due to the size, it also is one of the devices that has the lowest specifications in our catalog. So also when this one worked, we expect that many of the other ones worked. Plus, the EmStat Pico is made already for customization. So we made it as a module, so you can use development tools for hardware or use our software development tools to build something around it. So what we think is what we really contribute as a new possibility here to the market, to the field of field effect transistors based sensors, are portable versions, right? That you could take our EmStat Pico, build it in something portable that uses this field effect transistor technology. Right, and to do the whole thing properly, we joined forces. So PalmSens, well, it was as usual in life, just the right people talking to each other at the right moment in time. We talked to some people from the Institute of Physical Chemistry in Warsaw, which is part of the PAS. And well, and so we made a group for field effect transistors. So we had multiple people from the Institute of Physical Chemistry and some people from PalmSens. And we already had some success. So we wrote a manuscript that demonstrates how you can do these kind of characterizations. And we're trying to get it published right now. And we made an application note, which is already available on our knowledge base. Oh, and the paper manuscript is available as well. Right, so this is what has happened up to now. This is also why we have a guest speaker today, because part of this cooperation is Marcin, who is going to be the main speaker today and telling you about field effect transistors. Marcin, the floor is yours.
Hi, Lutz. Hi, everybody. It's great to be here. Thanks for the invitation. A little bit, we'll talk today about the electronic biosensors using FETs as the transducer. In the first part, we'll go through a little bit of introduction, and then we'll proceed to some applications about the whole thing. But let's start with a little bit of advertisement. So who am I? My name is Marcin Szymon Filipiak. I'm a Polish researcher working at the Institute of Physical Chemistry of Polish Academy of Sciences in Warsaw, Poland. My education and work experience are as follows. I've graduated from the Warsaw University of Technology with the major in microbioanalytics. And then I moved to Germany, to Heidelberg, to pursue my degree in physical chemistry at the Heidelberg University. Last but not least, I'm also right now doing my MBA studies back in Warsaw, Poland. When it comes to the work experience, well, during the Heidelberg times, I've been working in a startup company, sometimes marketed as the private research institute called Biomedics Institute. And our group was a pre-early stage development group for a corporate sponsor, in this case, Roche Diagnostics. From 2020, I've been back in Poland at the Institute of Physical Chemistry of Polish Academy of Sciences, where I'm holding the assistant professor position, and I'm trying to do my own research. So basically, a little bit more about the institute itself. So it's located in Warsaw, Poland. It's been founded already in 1955, so we have a pretty long history. And we do interdisciplinary research. Despite the name physical chemistry, we're doing chemistry, physics, as well as biology. And then we also have some material science capabilities here as well. There are more than 350 scientists working at the Institute, and there's more than 200 publications, peer-reviewed, obviously, a year. Now let's get back to our main topic of today's presentation, which is the field effect transistors. As probably know, field effect transistors are a basis of today's electronics. For example, let's talk about here metal oxide semiconductor field effect transistor, in short, a MOSFET. This is found in all today's electronics, and it can be used, applied as a switch, amplifier, or for example, a digital logic gate. So the electronic industry knows it very well, and basically how you're hearing me now is because of the transistors. Let's take a look into the structure itself. So we have three terminals, connections. There's a source, drain, and the gate. Let me pull out here. Oh, here. And a gate terminal, source, drain, and gate. And there is a semiconducting channel in between the source and drain terminals. And on top of that, here, the white layer here is the dielectric layer placed on top of the semiconducting channel. On top of this dielectric, there is our gate terminal. And now, in order to measure any transistors, any characteristics, we need to keep in mind that there are two potentials applied between source and drain, and between the gate and the source. So the metallic gate here modulates the charge carriers, either be it electrons or electron holes, in the channel via so-called the field effect. So in a nutshell, we can think of it as a in terms of the water tap analogy. So basically, our source, let's think of it for a second about it as the pipe. The drain is our drain of the faucet, and the gate is our tap. So what happens here is that the gate, the tap, controls the water flow flowing from the tap. Similar situation is here, but the gate is actually a potential applied to the gate. And with this, we can control the current flowing in the semiconducting channel. As I mentioned already, we need two distinctive potentials applied in order to measure any field effect transistor characteristics. There are two basically different approaches to measure them, and they're basically complementary, which means that you either do one or the second and you still get your information. So in here, what you can see is the EmStat Pico development board with the commercially available metal oxide semiconductor field effect transistor placed into the screw terminals. And then you can do two types of characteristics. Either you do the transfer characteristics, where the source drain voltage is kept constant throughout the measurement, and then you sweep the gate voltage in time, or you can do the output characteristics, which is the opposite, meaning that you change the source drain voltage in time, and then you keep the gate voltage constant throughout the whole experiment. And here you can see a very simple scheme how to use EmStat Pico Bipotentiostat in order to connect correctly the MOSFET. This is a very simple scheme. You can see it here that we're connecting the working electrode and counter electrode here to those two terminals, and then the third terminal, the drain, is connected here. This is easy as that. When it comes to the sole characteristics, how do they look like? Well, they look like this. So basically, in here you can see the transfer curves, transfer characteristics. So we have here the gate voltage for two transistors, one P-type transistor and one N-type transistor. This is a logarithmic scale here, so you can see that there's like several orders of magnitude of current changing from 0.1 nanoamp to milliamps even here. So this transfer characteristics, you can also think, as an electrochemist, you can think that the source drain current is constant here, so it's kind of like a current perimetry, and the voltage, the gate voltage, is swept, and it can be thought as the linear sweep voltammetry. And what we're measuring here is the source drain current all the time. Similar situation with the output curves. In here we have the source drain voltages in here, and here source drain currents for two different transistors, a P-type and an N-type transistor. And again, keeping the same analogy, the gate voltage here is in terms of current perimetry, whereas the source drain voltage is swept, so we can think of it as a linear sweep voltammetry. And what is measured is, again, the source drain current. Let's then take a look at how does our measurement, our results compare to the state of the art. So the state of the art is, for example, this Keithley source meter. It's a huge desktop piece of equipment, and our EmStat Pico is the one that Lutz already presented before. So this is a simple transfer curve in the linear scale here for one particular p-type MOSFET, measured with two different pieces of equipment, EmStat Pico and the Keithley 4200A. As you can see, both the curves do not overlap. Yes, that's a little bit, there is a little bit of difference. And here you can see the same plots as on the left-hand side, but now in terms of logarithm of the current. There is slight change in the potential threshold voltage, but apart from that, you can see the whole curve here. You also see here that the EmStat Pico is a little bit limited to about 10 nanoamps of lowest current, and this is already in the absence of Faraday's cage. So imagine that with the proper Faraday's cage, it could be even better. But also keep in mind that those two measurements were done by two different pieces of equipment. One, this Keithley source meter, which cost about 15,000 euros, whereas this EmStat Pico module, which is about 30 millimeters by 18 millimeters, costs about 500 euros. So for a fraction of price, you get maybe not perfectly same, but still feasible and doable characteristic measured with this piece of instrument. Let's talk a little bit more about the application of those FETs into biosensing, which is the core of our meeting here. In here, we have the so-called extended gate field effect transistor, where we connect the commercially available MOSFET's gate actually, to a conductive surface. In here, in this scheme, this is a gold electrode. And then we also immerse the reference electrode in the same electrolyte. So the absorption of some charged species on the extended gate here changes the IV characteristics, the current voltage characteristics of the MOSFET itself. The absorption, the charged species do not affect obviously the reference electrode because there is a FET. But whatever adsorbs in here on the this extended gate gives an addition to the surface potential, and that gives an addition to the IV characteristics of the MOSFET itself. In an ion-sensitive field effect transistor, the whole structure of the transistor is in the same solution, but the sensing surface here is the dielectric layer on top of the semiconducting channel. So the adsorption of the charged species on the dielectric here changes the IV characteristics in the of the field effect transistor, meaning that it changes how the current flows in the semiconducting channel. And then there is the third. Today, let's talk about the third exemplary architecture, which is the electrolyte gated field effect transistor, which is a little bit more advanced structure. We'll cover that more in the second part next week, same time, same place. In here, we use some fancy nanomaterial, for example, here, single sheet graphene, and then we gate we modify the IV characteristics of this current flowing in the graphene by directly biasing it through the liquid. So the adsorption of charged species on the semiconducting nanomaterial surface give a change to the IV characteristics. This one has also the biggest so to say, field to explore, meaning there can be lots of different effects, for example, Faradaic effects. So this is a very now emerging field, and people are pushing that forward, and I'm actually trying to push it as well. Today, we will cover mostly the extended gate field effect transistors, which in principle anybody can measure in their own labs, basically at this time even. So what we need is the commercially available MOSFET. We need the EmStat Pico module, and then we need some sensing surface and the reference electrode. So we made some basic study here where we use the glass electrode, so a pH meter glass electrode. This is actually a combined electrode, so it has two electrodes in one body. So there is one glass electrode, and then there's another electrode coupled to that one, which is the reference electrode, AGCL standard electrode. And this is used for obviously pH measurements. So we connected it as follows, as in this scheme. This is how it looks like in reality. It's not a very sophisticated system, but you will see that it still works. Again, the glass electrode, the glass light, the glass bulb is the extended gate, and the coupled AGCL electrode is used as the reference electrode here. And this is the method script that we used. So as you can see, there are 38 lines of code. At least 2 of them are blank. And then there's like 1, 2, 3, about 5 that are in every single method script code. So what you need to do is you need to set up about 30 lines of code, which in my opinion is not that long. And PalmSens themselves offer very good support for writing this code. So don't worry, it's not that scary. In any case, then we measured the pHs. So again, this is our more professional electronic scheme of our system. This is how the pH electrode was immersed in the buffers. Again, extended gate was used as the glass pH electrode, and the coupled reference electrode was used. And here is the main result. So let's go through it slowly. So on the left-hand side, the straight lines here are the responses from 3 different pHs. So we have pH 4, pH 7, and pH 10. And as you can see, the absorption of in here different concentrations of protons change the current voltage characteristics of the MOSFET itself. On the right-hand side, you can see the scale in logarithm, and we can see the same curves, but just in a logarithmic scale. Those are the dashed lines in here. What we can do in here is we can, for example, take some readouts. For example, for 1 microamp, we can read out the voltages in here. And then from those 3 voltages, what we can see is here, the dependence of the gate potential in those crossings here versus the pH. And as you can see, this is like 3 points here. This here resembles very much the Nernstian behavior. So what happened here is that the pH electrode, which is obviously sensitive to protons, is giving us the response in the Nernstian behavior as probably predicted. We can also make here a time-dependent measurement, for example, where we set up one particular gate voltage, and then we monitor in time the current flowing in the source drain semiconducting channel. Then we can perform some time-dependent measurements, and we can see in real time what is happening. This will be seen very nicely in here. in a comparison between field effect transistors and potentiometry, the classical, so to say, method in electrochemistry. Again, our scheme of the system. This is our scheme of simple potentiometry, where we measure the open circuit potential, and this is our real-time dependent measurement using the field effect transistor. So again, I measured here 3 different pHs, 4, 7, and 10, and simply moved the whole pH electrode from one bottle to the second and the third, waited for some time for stabilization, and then moved to the other one. What you can see in here is the change of current here. So from about 10 to the minus 7.5 currents here to about 10 to the minus 5.5. So we have 2 decades of current difference between one and the second step, one and the second concentration of our analyte. In classical potentiometry, we measure the open circuit potential instead. And then what we can see here is from about 150, minus 150 millivolts to about 25 millivolts, but on the plus side, so to say. So about 175 divided by 3. Why? Because there is like 3 decades of pH difference. We see about 56, 57 millivolts per decade, which again is the same as we measured in the field effect transistors. So to sum up, in here in the field effect transistors, as the current change here, or the transfer curve shift, or the analytical signals, but in here we measure the current instead of the potentials. And that's why we need a bipotentiostat to measure, actually to apply 2 different potentials and then measure current from the source drain. In the classical potentiometry, we measure the open circuit potential between the 2 electrodes, and this is our analytical signal. And the measured voltage resolution with this particular piece of instrument is about 45 microvolts. So the scale in here is in volts. The difference here is about 170 millivolts, and the resolution is 45 microvolts. Whereas in here, the difference is 200-fold change of current with the lowest resolution possible, about 0.006%, which effectively yields us more precise measurements. And actually, this was already noticed by the IUPAC, that the output analytical signal is effectively the same as the one, the same. So ESFET is the same as ISE, so ion selective electrodes. Lutz mentioned in the beginning that field effect transistors are sometimes not considered as electrochemistry. There is still somewhat a debate. I know professors who don't like to call them electrochemical biosensors. I like to call them the field effect transistor amplified potentiometric sensors. So we still use the potential differences. We still get the output signal in as a potential difference, but we're measuring current, and therefore we're more precise and can get much better analytical performance of our assay. Now I would like to present some more details about biosensing itself and why it has been challenging so far, and what was my colleagues and mine way to solve those issues. So, as I mentioned already, the key advantage of field effect transistor-based biosensors is the high sensitivity to surface charge. So the surface charge changes, then the surface potential changes, and that gives the change to the IV characteristics. But there is also a key challenge that is also known for the potentiometric field, which is the device screening limitation. So what happens here is that the surface potential of a given surface, given electrode, is in terms of this distance and this here, the bi length. So the bi length is this characteristic measure of how far does the sensor can see in principle. This is in a very basic terms. And this is also dependent on the ionic strength in and you can see that the dependence in this simple scheme. So in here, what we have is on the bottom, we have the electrode and some receptors adsorbed on the surface. We have here the two borders for concentrated buffer and for diluted buffer. This is this characteristic value of the bi length. In a concentrated buffer, the sensor can see only until this border here. So it is fairly easy to measure such small substances like protons or electrolytes or even DNA, which are very small and highly charged. For bigger entities, like in here proteins, and this is a scheme of a antibody, the binding events take place outside of this bi length. So in a concentrated buffer, all those events cannot be seen by the electrode itself. In a diluted buffer, yes, this is easier because again, this is dependent on the ionic strength. So how to overcome this limitation? Well, there has been several solutions that has been postulated to solve this issue. So we can use the desalting buffer or just wash buffer. So incubate our sample with the analyte and then just change the liquid, sure. We can do also some signal enhancement by label. So basically like a secondary antibody or a secondary substance that adheres to, absorbs to the analyte and then gives some signal. Or we can use some very easy solutions like shorter receptors or additional polymer layer. And I would like to focus here now on those two. So when it comes to the receptor size in here, immunoFETs, if we again visualize the sensor surface like this, our whole antibody can be about 15 nanometers in height. Obviously, it's just for the sake of this discussion, this is just a model. The antibody fragment can be about 7 nanometers. And for example, a nanomody, one of the newly discovered for the last 10, 15 years, receptor can be even smaller, about 4 nanometers. There has been a very interesting study where people were testing the whole antibody and then it's cutting into smaller pieces and immobilizing them on the surface. I will show that in a second. So why we do that even? Why is the receptor size important? Well, the reaction takes place closer to the electrode, and that means that the bi-screening is somewhat reduced. Then you can also achieve higher surface density of receptors. So instead of having big receptors on the surface, you get smaller, and then therefore there can be much more of them at the surface. And then the protein engineering, for example, can help you for specific and oriented immobilization. For example, if you make a fab with some free cysteine on one of the ends, then you can easily couple it via tile chemistry to the gold surface. I was mentioning this interesting study. So the researchers here took the whole IgG, whole antibody, did some pepsin digestion, achieved some Fab2 fragment, and then with some reducing agent, they cleaved it into Fab. Then they used the same surface immobilization protocol to compare directly what is the effect of the receptor size on the signal. And this is the result here. So what we can see is that what we can see in here is that for high PBS strength, so high ionic strength, the results, the analytical readout is much, much smaller for a full IgG, whereas for a small fab, which is, again, higher density on the surface and closer binding event, this is much, much higher. So if possible, it is recommended to use as short as possible receptors, as small receptors as possible. So same immobilization strategy, and then shorter receptors yielded higher signals. Now let's talk a little bit more about this surface modification with additional polymer, which is a pretty nice topic for the last couple of years. It was started by Gowell and Lieber in Harvard University, and then it spread out into other places. So basically what we've done in this study is that we immobilized on the gold surface some polyethylene glycol. We used so-called a mixed self-assembled monolayer. So we mixed a 0.5 kilodalton polyethylene glycol with some COOH groups. And then we also added some 10 kilodalton polyethylene glycol. Those are very hydrophilic linear polymers that you can easily attach to the surface with some tile chemistry. And this is a QCM study here. So what we see in here is the change of frequency, basically damping of the quartz crystal oscillations in terms of time. So after initial stabilization, we can see the monolayer formation here, exchange of the buffer, and activation with a very easy amino coupling procedure. So those COOH groups at the surface here get activated, and you can easily couple the Fab2 fragments that we had. In here, what you can see is the Fab2 immobilization. So again, the oscillations of the quartz crystals are damping because there is some more and more adsorption at the surface. And then there is some blockage with some bovine serum albumin. And then at the end, what we end up is to test is a ready surface, ready for testing some specific interactions, specific binding in here of thyroid stimulating hormone. As you can see, for low concentrations, this technique is not very feasible. Only with high concentrations, so like 30 nanomolar or here 264 nanomolar, we can see some adsorption at the surface. So what we see in here is that the Fab2 are covalently attached, coupled to the surface, and they are available for the target analyte, which is seen in here by this analyte. Antigen antibody interactions visualized in the QCM. And then the polyethylene glycol can reduce the non-specific adsorption. You can see that in here, after the whole immobilization, the whole surface modification protocol, we then added some BSA, high concentration of BSA, and there is no signal. The surface is saturated. Then we proceeded with some electrical, so field effect transistor based measurements. So we first tested the surface that the Fab2 fragments were immobilized on, but there's no this long peg, so we called it without peg. This is our readout, our real raw data, so to say. So those are the transfer curves shifted upon addition of some higher concentrations of thyroid stimulating hormone. And again, it was read out for a specific value of current at what you can see in here, that in low ionic strength, we can see some specific interactions with the TSH, and for high ionic strength, the black squares in here, there's basically nothing. The situation changed when we added additional this 10 kilodalton polyethylene glycol. Again, this is our raw data. And so the transfer curve so the source drain current in terms of the gate voltage. We tested several different concentrations along with the inert protein, here BSA. And then, as you can see here, the achievable signals are much, much higher. So mind that this is the same scale here, that this one. So the addition of this 10 kilodalton peg makes this highest achievable signal for highest concentration about 3 times higher than without the polyethylene glycol, which proves the point that the polyethylene glycol treatment, this 10 kilodalton peg, does work. Interestingly, I would like to also show you like continuation of this work. So we also measured some TSH in spike horse serum. And for that cause, we also used a concept called REFET, so reference field effect transistor. The concept is pretty simple. You take 2 sensing surfaces, one surface that is modified, immobilized with some specific receptor. In here, this is Fab2 fragments. And then you take another sensor that is modified or immobilized with a very similar protocol, but instead of the specific Fab2 fragment, you put here either the isotype receptor, or you put just like an inert protein. In here, bovine serum albumin. And what's done in here is that basically you get 2 signals, one from the active channel, so this one with a receptor specific to our analyte, and then you get the signal from the control channel, which is this one with this inert protein. And from that, you can get the specific signal stemming only from the actually antibody-antigen interactions. So this allows us to eliminate the biological matrix effects and the whole non-specific absorption, changes of temperature, drift, and so on and so forth. The 2 surfaces should react very similar, but one should be specific to the analyte, the other one should not. In here, you can see that we performed measurements. This is already a subtracted curve here. So with a blank, meaning that there's just the spike horse serum. After subtraction, the active and control channel, there's basically no voltage shift, no signal at all. Whereas when we add the 25 picomolar TSH, we can see that there is some potential change difference in here. So having this as a baseline, we can get a very, very precise and very nice measurement. What we then did is we also tested it with the against the state-of-the-art piece of equipment. In here, this was a COBAS system, a laboratory piece of equipment that Roche normally delivers to like medical laboratories, like hospital laboratories. And this is based on electrochemiluminescence. And we tested our method, this field effect transistor-based method, against their method, their ECL method. And as you can see here in the Bland-Altman plot, the results taken from our FET technique do correlate with the results that we got from the commercially available state-of-the-art diagnostic system. So with this, I would like to conclude for now. So what we got to know today is that the FET biosensing is the re-emerging field with a lot of interesting aspects. And that also EmStat Pico, the PalmSens product, is feasible for measurements of those FETs, and that the commercialization is definitely possible. But there's not so many examples of it. There is no point of care system on the market yet. So it is up to you to take up the challenge and then maybe come up with one. And for the next time, so again, same date, same time, same place, we'll talk more about multiplexing with EmStat Pico. We'll talk more about this single sheet graphene field effect transistors, which are the core of my scientific work. And also I had some side project with organic electrochemical transistors, and we'll cover that as well. And with that, thank you very much for your kind attention. I'd like to acknowledge my collaborators from the PalmSens side, Mohit, Lutz, and Brendan, as well as my Bachelor of Engineering student, Maria Wróblewska, that benefited from our collaboration with PalmSens in the form of her defended Bachelor of Science in Engineering thesis. Thank you very much, and I guess now it's time for discussion, I guess.
Yes, so thank you, Marcin, for this, for the great talk. Just one thing I would like to add, well, because you guys know I'm more from the PalmSens side, the method script was mentioned just for everybody who hasn't attended our previous webinars about method script. That's a scripting language that we have developed for our EmStat Pico, and for the EmStat Pico, for our EmStat 4, it allows you to automate processes with the potentiostat, but also actually take more control of the potentiostat by writing a rather simple script. And don't worry, you don't have to start from the scratch. PS-Trace, our regular user interface software for Windows, can generate a piece of method script for you, and then you can modify it to what you need. And there's a lot of documentation online on our website available. Well, don't forget to write your questions in the Q&A, and maybe I think there are actually quite a number of questions, so don't forget to vote for the question that you find very interesting. And maybe Adi already has picked his favorite question for us.
Yes, many people are asking questions. Also, don't forget that people you can vote for which one you like most. So now I'll see the top one has 4 votes, so we'll start with that. You will either have to see Marcin and Lutz for which of you this question is best for.
I can see the question as well.
This is one from Norman Pfeiffer. He asked, is it possible to use electrochemical impedance spectroscopy to measure transconductance with PalmSens equipment?
The short answer is yes. The short answer is yes, although for now, the solution for that would be to use 2 pieces of instrument. So 2 EmStat Picos. One would be the one that applies some AC voltage. The other one would be the one that takes the another DC voltage. Why is that? Well, for now, the method script doesn't allow to perform this AC voltammetry or EIS in the same in a bipod mode. So you basically need 2 pieces of equipment, one connected with the galvanic isolation dongle, and then the second one connected normally to his USB. So with those 2, using the multi-trace piece of software from PalmSens, and then adding some method script, you can definitely use this. And this is on my list actually to test next.
All right, thank you, Marcin. The next question is from Dimitrios Simatos, and he asks, so what's the PAG's role? What is the sensing mechanism, and are you sensing the analyte's charge?
Yes, so the sensing mechanism is based on the charge discrimination. So imagine that you have some charged molecules in the solution, and then it absorbs on your surface. So it can add up some charges, or it can also neutralize some charges of the surface that was initially charged already. So basically, the surface charge difference is what you are measuring effectively with field effect transistors, but not in a single point measurement like with potentiometry, but having the whole curves, for example, like those transfer curves. Or if you want to do the time-dependent measurement, you apply 2 potentials and then you monitor in real time.
All right, great, thank you. And another question is, does 10 nanoamp make sense in bio-wearables? I mean, using nanoampere seems good for academia, but are those nanoamps valid in reality?
Well, that's a very good question. So as you could see in my results, then 10 nanoamps limitation we recorded some noise basically there. But then the whole curve was still intact. So for a real world application, the EmStat Pico itself is good enough. You don't need to go to so low currents. You can stay with the higher ones. And it's a matter of selecting the correct MOSFET in order to get you the most out of the assay itself.
All right, that makes sense.
So so just to comment on that. So important is that the linear range, right? The part where your signal is proportional to the effect that you want to measure, that this part is clearly resolved and noise-free, right, Marcin?
Yes, that's correct. And then again, even if it's a little bit noisy, the normal procedure that I take for data analysis is that I normally just interpolate this linear region in logarithmic scale, so to say sub-threshold region with a linear regression, a function, and then you can read out for a specific current, you can read out more precisely by means of just data analysis.
Great, thank you. Lutz, this is another question for you. Can I use the PalmSens 4 for this experiment? And I guess this will be with a biopotentiostat.
So we have not tested the PalmSens 4 for it yet. So I don't have empirical data. However, it is a biopotentiostat and it is capable of doing the same things. There's just one obstacle that you will have, which will make the thing slightly more complex than with the EmStat Pico in that sense. So we have used in principle, the EmStat Pico could, through our Windows software PS trace, apply a linear sweep to one of the two working electrodes and apply a constant potential to the other one. And as Marcin has explained, this is what you need to do for output or transfer curves. The PalmSens 4 with a bipod can do the same thing. However, in our regular software, the bipod working electrode does not do auto-ranging, so it does not choose the current range for you. Marcin has now very cleverly used method script to basically write its own auto-ranging for the EmStat Pico's second working electrode. So in the setup that Marcin has shown here today with the EmStat Pico, the current that is measured on both working electrodes is using auto-ranging. The PalmSens 4 cannot do this at the moment because it does not support method script, and PS trace does not support auto-ranging for the second working electrode. However, a workaround that you can do is, well, one thing is first, maybe your signal is not going through that many magnitudes of current, and the PalmSens 4 current ranges are actually very, very broad. It's 6 times the current range value, meaning if you would be in the 1 microampere current range, you can go from -6 to +6 microampere, for example, or if you're in the 100 microampere range, from -600 to +600 microampere. That's one way of doing it, just choosing one very good current range. The other option is that you actually split the measurement up in 3 measurements, each of them with a different current range, for example, and you can do that in the scripting window of PS trace. So it was a long answer. If I want to make it short, is yes, but not as conveniently as with the EmStat Pico or the Sensit BT.
Great, thank you, Lutz. And then Marcin, a question for you, which is actually a combination of multiple questions I see here. So the question is about biopotentiostats and how to connect it to an FET. So would it be the same as connecting one potentiostat to the gate and the drain and the second to the RE? And should you short it or not to the source?
Well, yes. So basically, I think that the best would be if I just simply share a screen again and then go back to one of the slides before. Okay. So what we have in here, actually maybe here, there we go. So in here is this extended gate field effect transistor based measurement. We have here two channels of the EmStat Pico. Either you can view it as a bipotentiostat or a two-channel potentiostat. Basically, you have here the 0, 0, 0. So this is one channel. This is the second channel. And as you can see, one of the reference electrode and counter electrode is wired together. And then this is connected to the source terminal. And then the working electrode 1 is connected to the drain. So we're measuring the current flowing from here through here, let me pull up the laser pointer, from here through the MOSFET to the drain in here. And then our second potential is applied through the working electrode 1 to the reference electrode through the solution into the glass electrode or whatever sensing surface, and then back to the MOSFET itself. In principle, it could be possible to use two separate potentiostats, but then you need to synchronize them, and then you need to put them on the same ground. So to answer your question, yes.
Yeah, that makes sense. And this sheet is actually perfect as well for the next question, because on this sheet...
Wait, I need to say something to this. They would need not to be on the same ground. They would be galvanically isolated so they can share electrodes.
Yes.
So they would need to be floating, both of them. So for all of you out there who do have already, for example, one of our multi-channel potentiostats with galvanic isolation, it should work. We don't have empirical data. However, it should work using two channels to do that. Then you would need to, basically you connect both of the potentiostats as two electrode systems, one of them between source and drain, and the other one between, gate and source, or gate and drain, and then it should work. Should.
Yeah, Marcin, this sheet also shows another question that some people find confusing. The CE of channel 0 and the RE of channel 0 are short-circuited together. Could you explain a bit why you did that?
Yeah, well, we just need the potential. So the two electrode system, we don't need the three electrode system to be separated in here. That's why we shorted the CE and RE together. And they're all against the same here ground.
Yeah, no, that makes a lot of sense.
Also don't forget, because it's all electronics, there's like no concentration polarization or these kind of things where what you need usually to separate the reference electrode from the counter electrode, that the reference electrode is current free, because the terminals don't change under the influence of current.
Yep, great. Marcin, can you also measure polymeric electrical characteristics like conductivity, permittivity, and others?
Well, depending on what do you mean by polymeric materials, because for example, in organic electrochemical transistors, where we use, for example, P.PSS, yes, that's definitely possible to be measured, because we're still in this plus minus 1 volt range, and the plus minus say nano to micro amp value. So this is definitely possible to measure OECTs based on polymers here. I don't know if this answered the question, but I hope so.
We'll see in the responses. And can all three types of FETs, so you described 3 types of FETs, can they all be measured by the EmStat Pico?
And that's very cool, actually. Yes.
Right. That's
basically,
short. I like that.
basically, every single piece of FET can be measured. Just keep in mind that the EmStat Pico itself has some limitations when it comes to the voltage that it can apply and to the currents that it can measure. So this is between, as far as I remember, plus minus 2 volts when it comes to the voltage applied, and then between 100 nanoamps and 5 milliamps of current, as far as I remember. Lutz, can you verify that?
Yeah, so the maximum current is 3 milliampere, roughly. And yeah, so it's minus 1.7 volts to plus 2 volts as the full potential range, or the maximum potential, there's a dynamic potential range, etc. If you want to look up specifications and details to compare if it would work with your field effect transistor, I recommend to go to palmsens.com and look at the PalmSens product website, at the EmStat Pico's product website. There's a specification sheet. Then you can really look in detail for all different parameters if they would suit to whatever you're doing.
Just one comment that I came up with. So what I meant is that all the FETs that are measured and gated through the electrolyte. So backgated FETs where the voltages are like 50, 100 volts, not with this piece of equipment, sorry. But everything that is in solution, so like a reference electrode in solution or a pseudo reference electrode, that's fine. As Lutz mentioned, minus 1.7 to 2 volts. This is good enough for measuring any currents, any voltages in liquid solutions, because we don't want to end up having some water splitting, right?
Yeah, exactly.
While we talk about the specifications of the EmStat Pico, I think this is maybe a good point to point a special action out. Because we have, and I just quickly will share my screen for this. So we want you, of course, to try this at home. So we offer you a 10% discount on EmStat Pico development boards and the EmStat Pico Max 16. The later one will be introduced by Marcin in the next webinar. And you can get that discount for every quotation request that we receive before April the 16th. How do you get the discount? Well, oops, there we go. When you submit a quotation request, enter the promo code FET-webinar into the comment section before you send the quotation request. Then you will get a 10% discount on your EmStat Pico development board or your EmStat Pico Max 16. Okay, so I just wanted to put that out there before we forget that.
Great, thank you, Lutz. We're going to answer 2 more questions and then we're running out of time. I want to say to everyone that you can upvote your favorite questions, so we'll answer your 2 favorite ones that are still left. And meanwhile, Lutz, it would be great if you could answer the question, is this webinar recorded or not, and where can we find the link?
Lutz?
And otherwise I can answer it.
Yeah, sorry. I was just like getting with the hotkeys a bit. So, yes, maybe you've noticed there's a little red recording light running throughout this webinar, and you did get a little pop-up at the beginning of the webinar. Indeed, you all have been recorded and your wonderful active contributions. Thank you for all your contributions. And everybody who has registered to the webinar, so also your colleagues who couldn't make it today, but registered, they will receive a link to rewatch this webinar.
Yeah, and we will probably do that after we've done the second webinar. So we get you get 1 link or 1 email with 2 links in there. Great. There is one favorite one I see here on top. That's for you, Marcin. How does the PGA spacer help to improve the sensitivity?
Oh, that's a very good question. I like that question, and I'm obviously prepared for that. So this is a little slide on polyethylene glycol itself. It's a linear polymer molecule composed of oxygen and 2 molecules of carbon and then oxygen again. Those are the properties of the polyethylene glycol itself. So it reduces non-specific adsorption, stabilizes the protein structure, and it's also PEG effect in FETs setting is also now known and tried to explain. And this is based on the so-called Donan potential here. So for a solid electrode here on the left-hand side, the bi length the surface potential exponentially decays with the distance from the electrode. If you pegylate the surface or add any other like hydrophilic polymer, you make some kind of like a layer in that extends a little bit the solid. So the surface layer in here practically starts at the end of the polymer layer. So whatever happens in the polymer layer can be still sensed because the surface potentials are not changing that much with the distance. So again, here you have the solid electrode. Here you have the surface, this polymer layer until this line here, and you can see that the surface potential starts to decay much further, further away from the solid electrode, much further away from the distance-wise.
Right, thank you. And then the last question, what are the crucial parameters when it comes to down to choosing your FET transistors, like the N-type, P-type, the voltage?
Well,
what do you have to take care of?
Depending obviously on what do you want to do. When it comes to for example, this extended gate field effect transistors, where you use the commercially available FETs, what you should look for is the low threshold voltage. So you make sure that this is in the range of the voltages that EmStat Pico is capable of providing. And then also the saturation current of the field effect transistor, meaning that for a very like sophisticated power field effect transistors with high currents, with high potentials, this is not very feasible for this biosensing in here. You need to select the ones that work with low voltages and low currents and have as steep subthreshold slope as possible, because this is your readout. This you use as your analytical parameter, analytical signal.
Yeah, great. Thank you, Marcin. Lutz, I hand it over to you.
Yes. Okay, great. So I think this is it for today. I think we all got a lot that we have to digest till next week, where we will have a bit more a bit more modern field effect transistor, a bit more towards the border of what, more towards the border of what science is doing right now, right? So this was kind of the foundations towards an application, showing you how to do the measurement. Next week, we want to go into multiplexing, which shows you how to do it more efficiently, maybe, and make more miniaturized system. So Marcin has some self-made circuits there that he can show you as well. Yeah, so that's what we will do next week. For this week, I want to thank everybody who contributed here with questions or just giving us your time, so you would watch us. Also, I would like again to thank Ardi and Marcin for making this possible. So thanks everyone, and yeah, I wish you a great day and see you next week.
Yeah, thank you very much from my side as well, from invitation and for your kind attention. And should you have any more questions, don't hesitate to write me an email. I've sent it on a chat, but I think in the you can also contact me via LinkedIn, or however. I'm being also somewhat promoted by PalmSens. So thank you very much. See you next week then.
Bye.
Bye-bye.